Hello, everyone, and welcome to PAGBioDay. I know you all are probably just as disappointed as I am that we are not together down in San Diego for PAG, but I'm really excited to be able to host this half-day virtual event for us so that we can share and discuss all things plant and animal genomes. So we've got a really excellent lineup today. We've got 10 different on-demand presentations on everything from face mites to cannabis to dingoes. And all of those presentations will be available on demand all day long. So I encourage you to go check those out. In addition, we've got 2 live workshops hosted at 11:30 a.m. Concurrently, one focused on genome assembly and the other one focused on isoseq. So definitely drop in there to hear more about those particular workflows and to get your questions answered by the experts. Lastly, we have a general drop-in session that'll be open so that you can get any PacBio general questions answered by PacBio scientists. And then lastly, I'm actually hosting a live panel discussion at 12:15 Pacific with several of our speakers on the topic of the future of sequencing in plant and animal sciences. I imagine it's going to be a great discussion about how we tackle strategies with genome assembly and annotation, all the way into what we see kind of coming out in the future. So I encourage you to join us for that as well if you have interest. And we have a really great keynote speaker for you. But before we launch into that, I just want to reminisce a little bit about what we've done over the past year. So it was this time last year at PAG 2020 that I shared with you the HiFi sequencing data type and explained what all you could do with it. And really with the goal of convincing you that you should HiFi all the things. And throughout 2020, you did not disappoint. A quick scroll through Twitter shows just how much data you generated and all the really great science you did. And it's been really, really exciting to see how well HiFi is working in all of your hands. And for those of you who are newer to HiFi sequencing, here's just a quick explainer. So HiFi reads are generated by sequencing around a double-stranded DNA molecule that has 2 hairpin adapters on each end, creating a circular molecule. And as we're able to sequence around this circular molecule multiple times, we can stack up those individual reads and call consensus at each base. And that consensus called sequence is what we call a HiFi read. And these HiFi reads can be up to 25 kb or even longer. And they actually have a median accuracy of Q30 or 99.9% accuracy. And so with that read length and that type of accuracy, you could really imagine a bunch of different value that you can get from this data type across a wide variety of applications. So everything from genome assembly to high accuracy, looking at variants both at the single nucleotide level and at structural variation level, targeting really difficult to sequence regions, or getting full-length RNA transcripts in order to annotate your genome, or even parsing apart complex microbial communities in metagenomic samples. And with the continued increase in throughput that we're seeing on our sequencing platforms, most of these applications can be done in 1 to 2 SMRT cells. So it's really a cost-effective way to get at biological questions. And if we look at each of these applications, what I mean by a high quality genome assembly is that you're actually able to get extremely high contiguity. You are getting more of the genome represented within the assembly because you're able to get through repeats. You're getting very high base accuracy and making sure that all of the genes that you're getting are in frame. The HiFi data type actually has significantly smaller file sizes than noisy long reads, and that actually ends up reducing the analysis time. So you're reducing your overall project and project time and cost. And then HiFi data is actually pretty easy to work with because of the smaller file sizes and shorter analysis time. But also on the wet lab side, you're not trying to get extremely large DNA fragments because you're working in kind of this medium 20 kb range, which means handling of your samples is actually easier as well. And because we have easy end-to-end solutions, you're able to do the workflow pretty seamlessly. On the RNA sequencing side of things, the value of doing IsoSeq, which is PacBio's long-read RNA sequencing, is that you're able to get the entire transcript from the 5' end all the way to the poly A tail in a single read, so that you are not having to infer what the alternative splicing looks like across different mRNA transcripts, and you're actually able to just see the entire thing in a single read without assembly. When it comes to metagenomics, you can take a metagenomic sample from anything you're interested in studying, whether it be soil or water or some sort of tissue. And when you do HiFi reads, you're getting a lot of functional information because every read has anywhere from 7 to 9 fully intact, complete genes in it, so that you're able to resolve things down at the strain level. You're able to have more complete genome assemblies for things that can't be cultured. And you have very high contig purity. So it makes metagenomic samples really easy to work with as well. And on the targeted sequencing side of things, the high resolution and the long read length actually allow you to identify variants that you wouldn't be able to pick up with short reads, or to have a more zoomed in approach at local genome assembly. And so with all of that really great information on the various applications that you can do with HiFi sequencing, it was really cool to see some new things come out in the last year. So we demonstrated that you can assemble all things big and small. You might have seen that we actually tackled the California redwood genome, which is a 9 gig hexaploid. And with only 20-fold coverage, we were able to get a very contiguous, complete genome assembly. And spoiler alert, later today, Liz Sang will be presenting on the IsoSeq data that then came out of it, and the ability to use isophase in order to be able to phase the haplotypes. We also saw on the other end of the spectrum that we released the amplification-based ultra-low DNA input workflow, which really opens up the ability to make reference quality genome assemblies for really tiny arthropods. So that was really exciting. And there are several talks today focused on that work as well. We also saw an emergence of pangenome projects come out. And this is because not only has the cost and the time for generating genomes gone down, but the resolution with which we're actually able to look at the genomes because of the high accuracy means you're able to see the variation between individuals of an individual species. So there's a lot of work being done there that's really exciting to track. And then lastly, with the release of the Sequel IIe system, we're seeing that you can have better data with less time and more cost savings due to being able to get HiFi reads directly off the instrument, which significantly reduces your analysis time and your storage for data. And so really what that means is we have full end-to-end single technology solutions that help us tackle some of the big questions out there in biology, whether it's trying to breed drought-resistant crops in order to better secure our food source for the future, or protecting coral reefs from climate change and bleaching, to tackling diseases in our furry friends that might be used for translational research later, or protecting us from things like murder hornets, which are threatening to take out our honeybee population, or giving a little helping hand to some of the world's endangered species. There's really a way that HiFi sequencing can help in all of these efforts. And so with that little background on HiFi sequencing and how PacBio is helping conservation, I am very pleased to introduce Dr. Harris Lewin. He is a distinguished professor of evolution and ecology at UC Davis, and he's also the chair of the Earth BioGenome Project. And this is a large genome project that is aiming to sequence all species of eukaryotes on Earth, and is an umbrella under which many of the genome initiatives that you see out in the world, like the Darwin Tree of Life or the VGP I5K, are a part of. So he has a lot of really great experience in sequencing and understanding biodiversity. And so with that, I will turn it over to Harris.
Thanks so much, Michelle, for the introduction, and greetings to all of you. And thank you to PacBio for the invitation to speak on PAGBio Day. Today, I'm going to tell you about the Earth BioGenome Project, a little background on the project, and about the activities of the Earth BioGenome Project and its affiliated projects in the area of conservation genomics. Okay, so what is the Earth BioGenome Project? It's a project of confederated international network of networks that has a common goal of sequencing and annotating the genomes of all 1.5 million known species of eukaryotes in 10 years. Just a moderately ambitious goal. Now, the project as it stands, this network of networks, now includes 38 member institution network nodes on every continent except Antarctica. And its members are committed to the development of standards for the project, dissemination of best practices, and coordination of sequencing, analysis, annotation, and training activities. There's also a very strong program to develop ethical standards for participation in the project, data sharing, and access and benefit sharing. The International Scientific Committee of the Earth BioGenome Project is chaired by Stephen Richards at UC Davis, and the International Science Committee has 5 subcommittees chaired by internationally renowned scientists, each with specific responsibilities to core areas that will benefit from standardization across all the networks. And those are Mara Lonachak, who's the chair for the Sample Collection and Processing Subcommittee, Richard Durbin chairs the Sequencing and Assembly Subcommittee, Paul Fleechek chairs the Annotation Subcommittee, Shustin Lin-Blateau chairs the Data Analysis Subcommittee, and Xiaofeng Li from BGI chairs IT and Informatics. And all of the standards documents will be available on the EBP website. Some of them are already available, but all of them will be available within the next few weeks. Now, as I mentioned, there are 38 affiliated community, regional, and national projects in the that are aligned to the EBP. These are all aligned to the basic mission of the EBP, and collectively, these projects cover all eukaryotic taxa and represent most of the large-scale global efforts in biodiversity sequencing. And then what is the status of biodiversity sequencing? Well, of the 12 to 15 million eukaryotic species on Earth, including about 8.1 million species of plants and animals, there are only about 1.5 million eukaryotic species that have been described and documented and have been named. And less than 0.4% for a total of 5,700 species have been sequenced, at least at some level of whole genome sequencing. If you look at the level of the genus, about 2% of all 150,000 genera have been 2% of have been sequenced, and about 15% of the eukaryotic taxonomic families. Now, the reason you have that P1, P2, and P3 there is because those are the phases of the Earth BioGenome Project. So phase 1 is to sequence a representative species from each eukaryotic family. Phase 2 is for each representative species from each genus. And in the out years, years 7 through 10 of the project, to sequence all species. And so at a level of less than 0.4%, our current understanding of the evolution of life on Earth is extremely limited. Now, what about the quality of those 5,700 assemblies? This figure, which was beautifully made by Steve Richards at UC Davis, shows a plot of the scaffold N50 against the contig N50 for all the 5,700 species in the NCBI with genome information in the NCBI database. The genomes created with short reads mostly fall within the contig N50 of less than 100,000 base pairs and scaffold N50 with less than 10 million base pairs. These are the, let's see if I get my thing. Yes, these are these orange dots shown here. While those with long reads up in the upper right quadrant and high C scaffolding comprise the genomes with the highest continuity. Only the 410 genomes falling in the box, the orange box to the right, that's the shaded box here, represent genomes that satisfy criteria for reference quality genomes, while those in the yellow satisfy the criteria for the draft quality genomes. About 1,600 genomes in all in the yellow box. Now, important point is that assembly continuity is improving rapidly, and this is due really to the tremendous advancements made by PacBio in their technology, first with CLR and more recently in the past couple of years, and now brought to really to production level with CCS reads or HiFi. Okay, and this is just a little different way of looking at it. This is looking at these 5,700 assemblies across a 10-year time frame, and the left panel is all assemblies. And as you can see, we're actually approaching an exponential growth phase in the numbers of whole genome sequences for eukaryotes. And this is only eukaryotes. This does not include microbial genomes or bacterial genomes. But as you can see, the number of draft and high quality reference genomes is quite small. In fact, the number of reference genomes here on the right is only 7%. This is this 400 or so of the 5,700 total. Only 7% of the total are currently of reference quality. And this is important because as we move into phase 1, which is to sequence a representative species from each of the approximately 9,300 taxonomic families, we're going to be producing globally about 10 reference quality genomes a day, per day, every day, for 3 years to meet that goal. Now, the capacity to do that is really there today. And it's just a matter of coordinating it, organizing it in a way so that the right genomes are done in the right place and at the right time. And the exciting thing in looking at that figure on the right is, in green, is you can see this doubling of reference quality genomes in the last year to 400. Now, most of those genomes have been produced by EBP affiliated projects. About half of those are actually fungal genomes that have been produced by JGI, and that's for the 1,000 Fungal Genomes Project. And many of the others are coming from Sanger in the UK and from the Vertebrate Genomes Project at Rockefeller Institute, Rockefeller University, under the direction of Eric Jarvis. So these numbers are increasing rapidly and are going to really be exploding over the next 3 years as we go toward the goal of achieving 9,300 reference genomes in a 3-year period. All of this has been laid out in the article published in 2018, the perspective piece. And one of the stated goals in the perspective piece, and this is sort of my introduction to the conservation part of the talk, is the protection, conservation, and protection of biodiversity. And in that paper, we specifically stated, this is a quote from the paper, that we will sequence the genomes, characterize the genomic diversity of the more than 23,000 species at that time that are currently listed as endangered by the International Union for Conservation of Nature, and that this was going to be a high priority and a goal of the EBP. Well, things have changed in the last 3 years. The number of plant and animal species that have been surveyed by IUCN has increased to about 129,000 species. And what was then about 23,000 has now increased to 35,000, more than 35,000, actually 35,775 species are threatened with extinction. And that's 28% of all species of plants and animals that are surveyed. 543, it's been documented, 543 species have been lost alone over the last 100 years. And that number, 4,300, if you look at the fossil record, that number would normally take 10,000 years to accrue. So these extinction rates are well above the background rates of extinction and believed to be due to anthropogenic factors such as climate change and habitat destruction. And if you believe the models that are currently used to predict species extinction rates, the modelers, scientists have now predicted that by the end of the century, we may lose up to half of all plant and animal species due to changes in the environment and to the loss of habitat. Now, what about those species on the IUCN Red List? Carolyn Hogg at the University of Sydney has made this very nice chart, which shows that there of the 13,505 species on the IUCN Red List that are completely dependent on conservation, on management for their survival in the wild. Of those 13,000, which represents 38% of the species on the list, right? There were 35,500. 38% of those require today conservation for their survival. And of those, less than 1%, 0.8%, or about 100, have a genome sequence. So what is the value? Why do we want to sequence genome? Why do we need whole genomes in the first place? Many conservation biologists are happy sometimes with a bunch of SNPs or using RADseq. And this argument has been raging in the literature for a number of years, but there have been a few very important papers in the last few years, one by Beth Shapiro and Megan Supple, and the other by Kathy and Carolyn and Paris and Emma in Australia, that have really outlined why this is important. And the important reasons are, of course, with whole genomes, you can improve species definitions. Improving the definitions of species is important because it's species that you want to conserve. You can improve the distinction of where the boundaries of certain populations lie. That's very important for management strategies and maximizing genetic diversity between populations. You can reduce the level of inbreeding, and importantly, you can increase adaptability and resilience to climate change through the identification of alleles that improve resistance to drought, salinity, and to water tolerance. Now, this relationship between genomic diversity and threatened species was really elegantly illustrated by the EBP-affiliated project Zoonomia, the Zoonomia Consortium, of which I am a part. It was published just last month in Nature, and it has this beautiful figure that shows a plot of the overall heterozygosity of 7 endangered species plotted against segments of homozygosity. And as you can see, the 7 endangered species of the 131 new species that were sequenced as part of this project, the 7 endangered species had they did have significantly higher stretches and segments of homozygosity and overall lower levels of homozygosity. And nearly all of those species appear in that left quadrant of having reduced heterozygosity and longer stretches of homozygosity. And it's well known that species with lower genetic diversity have a higher risk of inbreeding depression and lesser ability to adapt to a changing environment. So therefore, genomic diversity is a very useful marker for understanding species at risk for extinction. Now, zoonomia is just one of 16 EBP-affiliated community, regional, and national projects with a strong conservation focus. I'm not going to be discussing all of these projects today. I'm going to be talking about two of them, and I'm going to be giving you some details about the goals and objectives and progress of two of these important projects. Now, all of the projects with conservation, not just the two that I'm going to speak about, share one important goal. And that is, as Carolyn likes to tell us, it's bridging the gap between geneticists and genome biologists and ecologists and practitioners of conservation, the ecologists and also policymakers. Now, as you can see from the slide, the gap here is very wide. It's still very wide. And there's a lot of research, training, and education on how genome information can be used to promote conservation efforts, because people on the right side don't quite clearly see the need for whole genomes just yet. But as I mentioned, this is starting to come as we begin to demonstrate the utility of having whole genome sequences for the development of policies and the implementation of these policies for conservation. And so bridging this gap between the genomicists and the conservation practitioners, ecologists, and policymakers is critical to lowering the extinction rates that I discussed previously. The first project I'm going to discuss is being conducted in Australia, which happens to have the worst record in the world, sorry to my Australian colleagues, but they know this, as a country for the extinction of mammals in the last century. Carolyn Hogg, Kathy Belov, and their colleagues at the University of Sydney and across Australia have set their sails to do something about it, based on their successes with the Tassie devil. Now, the Bioplatforms Australia, in collaboration with the University of Sydney and others, have launched a new program to bridge the gap between genomics initiatives and conservation management actions. This program aims to provide new genomic resources for Australia's and threatened species, reference genomes, associated population data, and an online portal that can bridge that gap between the genomic knowledge and those in the field who are implementing conservation strategies. The project that is just being completed by the Threatened Species Initiative is a pilot project. There's going to be much more in the future, but the pilot project involves the sequencing of 7 species, 4 of them are mammals, 2 are birds, and 1 plant species. All these species have been sequenced using PacBio HiFi for generating long reads, and all of them will meet the EBP minimum contig size of 1 million base pair standards. So this is being finished up, and the data for this project will be made available shortly. Second project I'm going to discuss briefly is the California Conservation Genomics Project, which is arguably the leading project in the world today, leading conservation genomics project in the world today. The project is led by Brad Schaefer at the UCLA LaCrette Center for Conservation Science, and the CCGP is funded by grants from the state of California to the University of California, with additional partners in the US federal government and private industry. The California Conservation Genomics Project will study 225 species that are distributed across 1,000 different sites in California. 130 distinct projects involving 71 principal investigators who are providing samples and knowledge about these species, where they're located and their biology. These 71 PIs are distributed across 10 UC campuses. As part of the project, 150 of the 225 will have reference genomes produced. And the 150 represent approximately all the genera in the 225 species. All of them will be sequenced with HiFi, and scaffolding will be done with OmniC. And this will all be done at the UC Davis Genome Center for the HiFi sequencing, at UC Santa Cruz, and Dovetail for scaffolding and assembly. And right now there are about 70 species whose reference genome sequencing and assembly in progress. The goal is to complete all 150 by the end of July of this year. Now, in addition to the reference genomes, what the scientists, the technical committee and the steering committee is also very important for the ultimate utilization of the data for this project is annotation. And to give equal time to all competitors, we must really thank Illumina for their program, the Illumina EBP 100 program, which has dedicated resources, flow cells, reagents, library kits for the annotation of 50 genomes. And this will be done by providing kits for library production, as well as the flow cells for sequencing both of the RNA-seq libraries and of the OmniC libraries. In addition to all of that, for the population genetic data, 150 species will have resequenced genomes. 150 individuals of the 225 species will be resequenced for population genetic data to obtain allele frequency data that will be useful in understanding the population genetics of these species and then ultimately used for management strategies. The figure on the left shows the state of California and the distribution of the 1,000 sites that are being sampled by the principal investigators and their students and colleagues. Now, the goals of the CCGB then are to have a broad and complete coverage of taxonomy of species in the state, of these, an understanding of these 225 species in the state, as well as their ecological coverage, and replicate this across for as many species as possible. And all of those sites, all of those species and sites are shown here in this part of the figure. Quite dense sampling, as you can see, for marine organisms off the coast from the border of Oregon all the way down to the border with Mexico. Plant species, vertebrates, and invertebrates as well. And again, as I mentioned before, similar goals to develop maps of this variation across the state, hotspots in the state that are resilient, places where you have resiliencies due to climate change, fires, as well as increase in temperature, salinity, and so on, to develop new policies that could connect the corridors to these hotspots to maintain genetic diversity, and recommendations to policymakers for the acquisition of critical lands to protect and conserve threatened and endangered species within the state. So much of the groundwork for the California Conservation Genomics Project has been laid at the UC Davis Genome Center with generous support from PacBio and an excellent collaboration with Jonas Korlak, who's been instrumental in supporting the Earth BioGenome Project and many affiliated projects around the world. I'm going to show you a few HiFi assemblies recently completed at UC Davis Genome Center that demonstrate the incredible power of the PacBio HiFi technology for generating highly continuous reference quality genomes. And all of this is done at a cost that we could barely even conceive of just 3 or 4 years ago. And I do want to congratulate PacBio on the tremendous advances in bringing high accuracy long technology to the genomics community. It has certainly been a real game changer. So the first species I'm going to talk about is the Western pygmy blue butterfly. This is North America's smallest butterfly and arguably one of the smallest butterflies, if not the smallest butterfly in the world. It is not an endangered species, but it does occupy some unique ecological niches in the state that make it an extremely interesting species to study. So this is a collaboration with Adriana Briscoe at UC Irvine and her colleagues, and Jonas, who was instrumental in providing, again, resources from PacBio for the sequencing and assembly. And as you can see from this genome, it's a relatively small genome. In fact, it's one of the smallest among the butterflies, or even the smallest, if according to Adriana, at approximately 215 million bases. I'm going to go rather quickly through these so as to save some time. But the contig N50, as you can see, easily meets the EBP standard on the primary haplotype. Each of these assemblies was done with HiFiasm and phased into primary and alternate haplotypes. The contig N50, 8.8 megabases and 7.3 megabase on the alternate, really extraordinary, as are the Busco scores with level of completeness, 99% completeness, complete genes in the primary assembly, 95.2% in the alternate assembly. Second species I'm going to discuss is an endangered species of butterfly, Baer's metalmark. Again, this is a collaboration between UC Davis Genome Center, PacBio, Earth BioGenome Project as part of the PacBio Earth BioGenome PacBio Earth BioGenome Project consortium. And the project is being led again by Adriana and Jonas at PacBio. Another excellent assembly with an N50 contig size of 4.42 in the primary and 1.61 megabases in the alternate assembly. Completeness is really, really good, 97.8% in the primary as far as gene completeness and 83.9% in the alternate assembly. Next species is critically endangered, the black abalone. At one point in time, the black abalone was the most abundant large marine mollusk on the west coast of North America, but now because of overfishing and withering syndrome, it has been much declined in its population size, and IUCN Red List has classified the black abalone as critically endangered. Now, Chloe Orland, who is on your program, you should be hearing from her sometime today, is likely to discuss the black abalone and the sequence, so I won't take up much of her time. But just to say, again, this is an amazing assembly, 14.3 megabase contig N50 for a genome size of just over 1 billion bases, with very high Busco scores in terms of gene completeness. Now moving to plants, you've heard about two insects, a mollusk. Now plants, this is a different collaboration also done as part of the PacBio EBP program. Again, a collaboration with UC Davis Genome Center and our collaborators at the university, at Unicamp in Brazil, the University of Campinas, and Embrapa. Isabel Gerhardt at Embrapa and Paulo Ahuda at the University of Campinas. And this is a great improvement over the existing assembly. There had been an assembly for this species, but Valosia are a fascinating species among a group of plants that exist in the Campo Rupestre region of Brazil. These are sometimes called resurrection plants because they look like they're completely dead. Their leaves turn brown, but the leaves are actually not dead. As soon as the rains come, these same leaves, they don't die and fall off and new leaves regenerate. They again turn green, and they're called resurrection plants. And this whole biological process is enormously interesting and under investigation by the group at Embrapa and Unicamp. And great assembly, again, exceeding the, this is a complicated genome with lots of repeats and other things, which has so far interfered with creating an alternate assembly greater than 1 megabase, but we've achieved 1.7 megabase contig N50 for this genome and very high BUSCO scores for both assemblies. The last one I want to talk to you about is the large tree shrew. And this one is has been done in collaboration with Oliver Ryder at the San Diego Zoo, who's provided the sample for this, Ollie and his staff at the Frozen Zoo, and Dovetail Genomics and PacBio. Now, this one is a completely scaffolded assembly. Some of the others are either in process or have been scaffolded. I haven't shown you the data, but this one has been scaffolded at UC Davis. The lead investigator is a postdoc in my lab, Joanna Damas, and the assembly has been done using the abbreviated version of the pipeline shown on the lower right side, Falcon Unzip for the HiFi data, SCAF10X for the linked reads, and Dovetail has done HiRise for all the data sets using Hi-C. And as you can see, the scaffold N50 here is 7.4 megabase just for the HiFi contigs. And when you add in the scaffolding, you've got a really high quality chromosome scale assembly at about 113 million bases for N50. Important to also note here that the L90, which is the given set of contigs, basically each of its own length, the L90 is the count that is defined as the smallest number of contigs whose length is the sum that makes up 90% of genome size. So 27 scaffolds make up 97% of the genome size, which is very close to the number of chromosomes. If you see in the upper right-hand corner there, you see that the chromosome number is 28. And so we're very excited about this particular assembly. And what this assembly allows us to do, is my last data slide, is fill in a missing part of the Eutherian tree. So we already have, for our work on chromosome evolution and ancestral genome reconstructions, at least a scaffold based to a chromosome level assembly for 18 of the 19 Eutherian orders. The Scandentia, the species which is represented by the order which is represented by the large tree shrew, was missing. And it's a very interesting order because its phylogeny has not yet been resolved. So having the sequence of this species will help us to understand where to place. It's really the last group of organisms, the last order that is not clearly resolved in the mammalian phylogeny. So we'll be able to resolve that issue as well as provide a detailed reconstruction of the ancestral mammalian genomes representing all Eutherian orders and most of the marsupial and monotreme orders as well. Okay, so to wrap it up, I'd close by returning to Carolyn's conceptual slide, which I think is useful. We are now acquiring genomes for endangered species at an extraordinary rate. The EBP and its affiliated projects can serve as an important role in bridging the gap between genome scientists and conservation scientists and policymakers to give them better tools for decision making. This is our responsibility to bridge this gap, to make sure that those who need this information or who will use this information for the purposes of conservation have the information in a usable form that can help these programs to advance. We expect that these programs will have a direct impact on conservation by slowing the rate of extinction of endangered species of plants and animals, as well as other eukaryotes that underpin the biosphere. So I want to close by thanking everyone. Go forth and sequence. Thank you very much. And now I'll turn it back to you, Michelle, for the rest of the program today. Thank you.
Thank you so much, Harris, for that wonderful talk on the EBP work. You're doing such great work, and I can't wait to see the progress as the years go by. As a reminder, we are hosting a live panel discussion at 12:15 Pacific, where we'll be discussing the future of sequencing for plant and animal sciences. So I encourage you to join us for that. In addition, you will have the opportunity to take home a thank you gift for participating in this first ever PAGBio Day. In order to get your gift, simply go to one of these live sessions that I have listed here, or attend more than one of the on-demand sessions. All of the eligibility requirements will be determined on the back end. You don't have to do anything beyond that. And you can look out for an email from us with instructions on how to get your take-home gift. And last but certainly not least, we are giving away free HiFi sequencing with our SMART Grant program. So if you have a collaborative research project that you're interested in doing, I encourage you to go apply for our SMART Grant. Submit your proposal by the end of the day on January 22nd, which is coming up rather quickly, but head to pacb.com/smartgrant for that. And with that, I just want to say thank you all so much for joining us on this exciting PAGBio Day. And please feel free to get in touch with us directly at pacb.com/scientist or check out our website.